I will go ahead and start. There may or may not be slides. In the event that there are not slides, we'll be okay. So what I was going to talk about is essentially how we make sense of single cell data using a process called cell type deconvolution, and when it doesn't work, which is, in my experience, most of the time, although some of y'all may have a different experience. So the way this process works, essentially, imagine you've got single cell sequencing data, which gives you, I think something about my PowerPoint the world just does not like. I'm sorry. Imagine you have single cell sequencing data, and then you have a bunch of bulk RNA-seq data from a bunch of people. What you could imagine is that you could somehow take that single cell sequencing data, identify cell types and gene expression in those cell types, and then if the bulk data is really like single cell data put in a blender, you would then be able to estimate across a population what cell types were present and in what abundances for each person. And you would imagine this seems really straightforward. This should just work, right? Essentially, you're taking a sum and you have the things that, that go into that. You should be able to just deconvolve that with some sort of standard linear approaches. Oh, okay, we're getting closer. Wonderful. There might even be slides involved in this presentation, which is going to be totally radical. In our experience, that doesn't always work, and it doesn't always work for quite a few different reasons. I'll talk about, in our hands, some stuff that does work, as well as places where these methods break down. If you want the punchline first, I think the biggest way that these methods break down is it turns out the gene expression that you measure from single cell data is not always the same as what you measure in bulk data. And it's not the same for quite a few different reasons. So one, a lot of stuff kind of goes to technical factors or various technical artifacts. This could be things like, well, it turns out that most of the bulk data that we get is from samples that have been stored in through what's called FFPE, or formalin fixation and paraffin embedding. Well, so it turns out that when you do that, you cross-link the nucleic acids to other molecules, and so when you try to reanalyze them, they break. They're broken. And so the way you actually isolate RNA from that is different from the way that you generally... Oh my gosh, there are slides. Rock and roll. Cool. Oh, thank you. I will make sure that whoever's I will not take your laptop at the end of this. I promise I will not take your laptop. I'm not 100% sure, but I'm going to try. Okay. So I'm going to... We wrap up at noon. Is that right? Noon. Okay. So this is the ideal world. Essentially, I've... A tiny bit past noon. Okay, so you start with single cell RNA-seq. You probably get that on a few people because it turns out it's really expensive. You could theoretically get cell type reference profiles out of that. And then if you have bulk sequencing, probably of many people, what you'd like to have at the end of the day is for each person an estimate of the cell types present in their sample that you took from them. We work a lot on cancer, so this is going to be in the context of high-grade serous ovarian cancer. Does anyone work in high-grade serous ovarian cancer? Okay, we will not go into the details of the disease. I will save time on that. I'm going to skip most of this just because we took a little while to get started. The one thing I will say is there are nominally a bunch of subtypes of this disease defined by TCGA and others, maybe 15 years ago now. If you think about subtypes, does anyone work on cancer subtypes? A couple people vaguely nod their heads. Do you work on like cancer subtypes, cancer transcriptomic subtypes? There's some people vaguely, so usually what we think about are like distinct entities, where if you look at some data type, in this case, the way these were defined, gene expression, if you look across a large collection of people, you would identify different clusters in those across a large collection of people. We would sometimes say those are subtypes. There's lots of different, if you've seen one cancer subtyping, you've seen one cancer subtyping. Ovarian cancer is not like cancers where there are radical differences between the subtypes. In this case, I would argue, although there are people who used to disagree with me, although fewer now, that these are more like a continuum of essentially whatever we're measuring here is not like a bunch of distinct molecular entities, but it's sort of like regions of density in this space that actually is relatively, also has kind of occupancy throughout the space. So I don't think it's like breast cancer where there are certain subtypes that are separate, and what makes them separate is individually targetable. This is going to be different and more complex than that. The only thing you should take away from that, though, is that there has been some suspicion for a while that these subtypes in ovarian cancer are driven by the tumor microenvironment. And so that would take you into the world of single-cell sequencing, which is where this story starts. OK, so when we worked on this project, so this is work that I did with Stephanie Hicks and Jen Doherty, and we're just wrapping up our R01 on this now. The idea was really simple. We're just going to do the exact thing that I told you about. We're going to take single-cell data from a modest number of individuals, bulk data from a lot of people, and then we're going to use that to estimate cell type composition in a lot of people, and then try to connect this back to both the subtypes and survival. So that's the kind of experimental design. We also were the people who asked, does it actually work? Like, let's test our assumptions instead of just making them and going from there. So this was work led by Ariel Hippen, who was a PhD student in the group, and she designed at the beginning a very simple experiment to help us figure this out. So we have a tumor. We are going to rRNA deplete that and bulk RNA sequence that, because we know that most of our data is coming from storage situations that require basically that we use rRNA depletion to identify the mRNA. So if you think about transcriptomics, there's, most of the transcriptome is really like rRNA, if you think about the bulk of what's in a cell. This, we're basically going to try to subtract that out, and that's generally how people work with archival samples. The other thing we're going to do is we're going to take those tumors, we're going to dissociate the cells, and then we're going to do single-cell RNA-seq. The most popular panels to do this are based on poly-A selection or some other kind of selection method that has the same biases. So essentially, in this case, what you do is you're essentially grabbing the tails of the mRNA. And so it's different. Instead of removing other stuff and measuring what's left, you're grabbing the tails and measuring what you get. And so this actually has an impact that could theoretically impact deconvolution. Normally what people do is they assume these are the same, and then they just apply deconvolution across both of these. So almost every paper that you see in the field that's sort of doing deconvolution is essentially taking archival samples that were probably processed with RNA depletion and assuming that you can create those by some additive combination of what you see in the single cell.
Sorry, can you go over depletion just one more time?
Yeah.
I feel like that's important.
Yeah, so you have all the RNA in the cell, and most of it is ribosomal RNA. So it's essentially structural RNA. And so what people usually do is, because of the way archival samples are stored, almost all the time you're basically pulling out the structural RNA and then sequencing what's left. So this is called rRNA depletion and then RNA sequencing. If you do single-cell RNA sequencing, your cells basically the RNA is not broken. And so the easiest, fastest, cheapest way to do it is just to grab the tails, which are poly-A tails, and then grab those and then sequence what you get. And so there is a true distinction here, and it does create some issues. Some of them are minor, but they can become kind of more painful. The other thing that we wanted to do was just assess pooling, because there's a lot of papers on pooling and a bunch of different ways to do it. In the context of ovarian cancer, I said I wasn't going to go into details, but there's specific reasons that pooling might not work here. And it's that there's basically a ton of focal CNVs. And so certain pooling strategies rely on genetic demultiplexing, and it wasn't inherently clear that those would work here. And so we took the same samples and dissociated them, barcoded them with cell surface markers, and then sequenced those as well. And then we also figured we'd round out the experiment by also doing bulk RNA-seq of the dissociated cells using the same technology that we use here, and then mRNA enrichment, and then bulk sequencing there. So we did a bunch of bulk sequencing, largely because you can do it for cheap. And if you're going to design an experiment that assumes something is true, you should probably check to see if it's actually true in your setting from time to time. So pooling, I am going to just briefly mention this. There's a couple different ways to pool. So one is essentially take your samples and stick something on the surface of all the cells in each sample that has a barcode on it. Usually you would use an antibody. There's some other strategies, but usually you'd use an antibody to some kind of ubiquitous cell surface protein. The alternative is, because these are samples from different people, you can use the naturally occurring genetic variants. So for every cell you get out, you would call variants in that cell, the measurements from that cell, and then you would try to use that to then assign cells back to people. So this is called hash demultiplexing. This is called genetic demultiplexing. We did both because we wanted to see how it worked. The punchline for this is essentially for the things for the cells where there was agreement, they work the same. The hash demultiplexing was less specific. So if you are working on data in this space and you have natural genetic variation between the things that are pooled, which would be true probably if you're working on human samples from different people. Not true if you're working on like mouse samples from the same strain of mouse. We found that genetic demultiplexing was probably a preferable way to go. Hash demultiplexing usually works. We were not able to attribute failures to certain cases, but it's not like random. It looks like for some samples you get less good antibody binding, which isn't the end of the world. I will say the other thing that's a little bit painful is sometimes it is cell type specific. So the antibodies bind less to certain cells and certain samples. So that's kind of worth knowing because it's not always advertised that way. But if you have naturally occurring genetic variants, my advice would be just skip hash demultiplexing and just use genetic demultiplexing. It seems better in every setting we've tested it in. OK. Yeah, so I'm going to skip this just because we got started a little late. Let's see, so dissociation. So dissociation actually matters. So we're going to compare these are going to be the same RNA prep. We're just looking at the dissociation markers. There's a couple different things that come up. So this is upregulated in the tumor chunks. That's going to be upregulated in the dissociated cells. The first thing to know is you're going to see a lot of differences in red blood cells. This is not terribly surprising. The dissociation protocol strips the red blood cells, so that's where those go. There's some, the other thing that we see is, this will not surprise folks probably, but adipose genes, much higher in the tumor chunks than the dissociated cells. So essentially you're breaking the adipocytes when you many of the dissociation protocols will actually break the adipocytes. So this may not matter so much for some tissues, but if you were, for example, working in ovarian cancer and you wanted to do deconvolution, the portion of reads in your bulk sample that come from adipocytes, you are likely to miss because you do not have them in your single cell reference profile. And so what does that do to your deconvolution method that is assuming that the bulk tissue is a sum of everything you see in your single cell? It depends. And so we see pretty variable performance over methods. And then there's some biology for this that I will, again, skip. And then the other thing that we wanted to look at, which I will briefly mention, is this mRNA enrichment. So do you get it by basically grabbing the tails, or do you get it by removing the stuff that's structural RNA? And here, this is a pretty big effect. So there's the bad news is that it is large effects in certain genes. The good news is in certain genes. And so there's likely ways to correct for this pretty easily. Some of these are really well documented. So the stuff on the left here, I will say, is poly A enriched. The stuff on, wait, is that wrong? Yeah, the right is poly A enriched. The left is ribodeplete enriched. So there's some stuff here like histones, for example, which don't have which actually don't have polyadenylation. They have a different mechanism that keeps that RNA stable. So it's a nice control. Let's see, do I have, yeah. And then mitochondrial RNAs are also different. But I guess one of the takeaways is if you're going to make the assumption that the single cell data matches your bulk data, there are large numbers of differences here that are driven just by the isolation protocol alone, the way you get the RNA. And so again, for your kind of methods that are going to assume additivity, this is going to be a challenge. Let's see, yeah, and then there's some other non-polyadenylated RNAs, some of which, like Turk, are actually really biologically important for ovarian cancer. We won't go deep into ovarian cancer. Another thing that's worth knowing is that a bunch of the protocols actually have their own unique features. So this is not a huge deal most of the time, but every once in a while people will use mitochondrial read count as a way to filter whether their single cell data is any good. If you're going to estimate what fraction of mitochondrial reads you should see from your bulk RNA-seq data, which sometimes people think is a good idea, you should know that certain protocols will preferentially and specifically remove mitochondrial reads, or mitochondrial RNA. So when you do the read count, you'll be vastly off. So for example, you might, if you use ribo0 gold, you might end up assuming that your total mitochondrial fraction in a viable sample should be under 1%. But if you use that to then set your thresholds for your single cell data quality, you will be in trouble because this is actually just what's getting, this is specifically removed by the ribo0 gold protocol that is widely used. And so again, caution, the way your data is generated actually does matter in terms of how you end up interpreting your results. Okay, so one of the things we then wanted to check was how do deconvolution methods perform in the context? Like let's say we assumed that these two things were identical, even though I've just shown you there are vast differences between them that you need to be aware of. How well do methods perform? We did this in a couple ways. So one, we can measure accuracy just by essentially doing the same thing that most folks do when they measure accuracy, which is take data and then create synthetic bulk data and evaluate how well you were able to generate the underlying data from your synthetic or pseudo-bulk data. So that's widely used. The second thing we did is, well, we said, well, we've got a bunch of different experimental conditions here. And the underlying composition of that sample is not at all dependent on the experimental conditions, right? It is, the composition of the sample is some true thing. Doesn't matter what the conditions are, but that is the actual thing it is. And so what we should be able to do then is ask which methods are stable over a bunch of different inputs, because conceivably any of those could be inputs to your deconvolution approach. And when we do that, we can measure, so first accuracy. So here you want the thing to be near 0, so ideally here-ish. And this is broken out by different methods. I didn't want you to compare methods, so I made it really hard to compare methods on this. If you look at the paper, there's a hard comparison or an easy comparison version of this. But essentially, the takeaway here is many methods are pretty close to 0. There are some that are kind of off. Some of them might actually be off. Yeah, I'll skip that. Too much nuance. But many of the methods kind of do well by the traditional evaluation metrics. It's not surprising, since that's how many of them are developed. The second thing we wanted to ask was how robust the results actually are. So here, lower is actually better. So it's the log variance. And so what we'd like is methods that were kind of accurate on the previous one and then robust. So regardless of what the input is, the method seems to produce approximately the same output for, regardless of what reference was used for input, the method seems to provide roughly the same output. And again, one of the takeaways is this is almost never used for evaluation of methods, but if you're actually going to apply these methods in practice, this is kind of what matters. And so we would make an argument that this is probably worth also considering. We ended up, for the rest of this work, we really liked a method that is now called Bayes Prism. It was originally published or preprinted under a different name, but it seems to have pretty good performance across a lot of the stuff that we have looked at in ovarian cancer. I would say that if you're going to run a large set of experiments in a different context, you probably want to do these same types of assessments. It's not terribly expensive to run that quick pilot experiment with a whole bunch of different bulk sequencing, and it could save you a lot of headache down the road. So here's the quick summary. Demultiplexing works. The loss of cell types is actually a pretty big issue. We think this is one of the biggest issues. I think one of the reasons Bayes Prism actually works has nothing to do with the kind of nice methodological parts of the approach. What it essentially does is it has one cell type that's much more flexible than the others, which is ideally the cancer cell type. And so that method can essentially, for all intents and purposes, suck up variants that would otherwise be an issue. Nominally, that's because cancer cells differ in their expression because different genes are mutated, so there's different kind of pathway activities. I think what it's actually doing is it's when there are missing cell types, like adipose cells here, I think it's essentially sucking all that variance together and preventing it from impacting the rest of the deconvolution. So it works really well, but probably not for the reason it's supposed to. The RNA isolation differences are also pretty big. There's a few different ways to deal with them. I will skip, let's see. I am probably going to skip this just in the interest of getting to the final part. I will skip this. Okay. So this is the last bit. This is a paper that Adriana Ivich, who's also a PhD student in the group, published the last few months. It was preprinted around the time of CGSI last year. Where she wanted to ask, well, with these missing cell types, is that signal somewhere present in the data so that you could recover it and potentially use it for deconvolution? I will note, our goal in this is not to build the method. We just want to ask the question, like, is that signal present in the data such that it could be used? And so this is how she ended up approaching the question, which is essentially trying to create a number of experiments where we've essentially removed a cell type from the data, and then we've asked how well methods can recapitulate it And there's a couple different ways to think about this. One is with single nucleus RNA-seq data. So there's a difference here from the bulk, which is that now you've only got nuclear RNA, but you've got it for all your cell types. So if there's something going on in the cytoplasm that's kind of specific and not a linear factor over what you've done, what's happening in the nucleus, that will be an issue. But on the other hand, you have all your cell types present. In the other bucket, you've got single cell data where you've got cytoplasmic and nuclear RNA. The challenge is you lose certain cell types. And so is there a way to use these two together to design an experiment that would let you test whether methods can actually be performant in this setting? The focus here is on adipocytes, which, so this is single cell, single nucleus, essentially is a cell population that's almost essentially completely lost. There are some others like the mesothelium that have the same thing happen. Okay. We've got 3 minutes. So essentially what she's going to do is she's going to take her single nucleus data as part of the experimental design, treat that as ground truth, but when creating pseudobulks, and then use the single nucleus data again as a reference, but essentially remove a cell type from the reference. So this gives us the ability to create, theoretically, what single cell data would look like in the context of one or more missing cell types. And so when she does that, we can then apply, so this is the experimental design. So we can then apply deconvolution to that. So basically take a pseudobulk, single cell reference, do deconvolution, you get your calculated proportions. And then one of the things we can do is go back and take the original pseudobulks, take the calculated proportions in the single cell reference, essentially multiply those together to estimate what gene expression is actually being explained by your deconvolution methods. Subtract that, get your residuals, and then what she's going to do is factorize those residuals. The idea being that maybe there's information in those residuals that reflect the cell type that's in fact lost. And if that is true, that would give you the ability to then create deconvolution methods that might be able to identify essentially structured, important variance in the data that is not being modeled in your existing reference and give you the ability to somehow feed that back in. She applied this over a bunch of different methods, a bunch of different experiments. OK, so we're going to skip to the takeaways so that I wrap up on time. So I think the first thing is the same limitations that I mentioned from Ariel's work at the beginning on the RNA-seq technology, so those same things are going to propagate through. So those same issues continue to be at play. The deconvolution methods that exist, when we do this experiment with these intentionally missing cell types, we see kind of some interesting failure modes for those deconvolution methods. And so I think part of the reason that's the case is that often when we build a method, we sort of assume the data will have all the signal in it in some form or fashion, and the key is just to find that signal. I think these missing cell types for most tissues are small enough that they don't swamp method's performance, so the method still looks pretty good. And because they're absent, we haven't generally taught ourselves to think about what's absent and then go see if the method is, erroring out on the things that we can't see. Right? It's pretty hard to design an experiment in that case. So by constructing the experiment where we then make things absent and then go look for them, we were able to kind of identify this failure mode for methods. I think there is an opportunity for new methods development in this space. And then I think it's just always important to be keenly aware of the constraints. This was quick and dirty, simple R01, should have been real fast. We should have been done with the analysis like 6 months after we started the grant, had it in place, and then just been able to add new samples and get better and better estimates. It's turned out for the entire 5-year duration of this grant, we have had to really poke at the methods and understand when they are and aren't working, because otherwise they give us results that are kind of biologically implausible. Like there are some papers in this field that apply deconvolution and that say that like 20% of a tumor is T cells. I am going to guess that most ovarian cancers are not 20% T cells, for example. Like that's an implausible cell fraction. And so I think the reason that's happening, at least in this field, is because the methods are not working. The assumptions the methods make do not actually apply. And so at the end of the day, that turns out to matter. It's probably different for other tissues, but I would say if you work in a field and you're seeing these methods applied and you're seeing cell type abundances that are kind of implausible with respect to the underlying biology, you may be seeing the same exact phenomenon in the data that you're working with. Okay, and this is available. This actually got published in Genome Biology earlier this year. Yeah, I was going to put in, we have a follow-up that kind of digs into that a little bit more, but if you stay tuned to what the lab's working on or you look at our GitHub, you'll probably find it right now. With that, I want to thank the folks who made this possible, folks in the lab, our collaborators, and then the samples that we ran this kind of detailed experiment on. We had to get a bunch of different tissue from a bunch of different places, and the Penn OCRC team was incredible for helping us get exactly what we needed for that. So thanks.